Hello, everyone, and welcome to Bioconductor 2021. This is a single-cell genomics short talk series session, and we're going to start the talk shortly, but first a few housekeeping items. If you have questions for the speaker, please enter them into the Q&A tab. Please clarify who your question is for, and we'll take questions at the end. You also have the ability to upvote questions for the speaker to answer so that we can kind of rank them. If you would like to ask your question live, please use the raise hand feature, and we'll bring you onto the stage so that you can talk to them. There are a variety of emoticons available to give feedback to the speaker as well. And if for some reason you have to leave early, a video of this session will be available a couple of hours after the session has been completed. So since we do have four talks during this 50-minute session, we likely won't be able to get to all the questions. But if you don't have time to get your question answered, then feel free to message here on AirMeet one of the speakers, and you can ask your question or potentially set up some future time to meet up for a longer conversation. For now, we're gonna go ahead and get started, and it's my pleasure to introduce our first speaker, which is Atefeh Taherian. She's a postdoctoral fellow at University of Queensland, who will be discussing cellular senescence through mesenchymal stem cell research.
My name is Atefeh, and I'm a postdoc at the Australian Institute for Bioengineering and Nanotechnology at the University of Queensland. I'd like to thank YC team for giving me the opportunity to present my work today. I'll be talking about unlocking insight into cellular senescence through single-cell transcriptomics of aging mesenchymal stem cells. This project has different exciting avenues, but I'll be presenting only the data analysis or bioinformatics part of the project. Also, this talk isn't really about a specific R package, but rather the utility of different R packages to analyze high-throughput single-cell RNA sequencing data. Cellular senescence is a state of permanent cell cycle arrest as a cellular defense mechanism against unnecessary damage induced by different stresses, including wound healing, cancer, and also aging. Senescence is one of the main contributing factors to aging and aging-associated disease. Senescence has also a complex phenotype. It is important to realize that bulk mRNA measurements of cells or even tissues might mask the heterogeneity associated with a complex phenotype. So using single-cell technology, we're able to study such phenotypes, and this technology has been widely used to investigate the impact of heterogeneity on cell fate. So here is single-cell RNA sequencing of mesenchymal stem cells undergoing replicative senescence to understand the heterogeneity associated with the senescent phenotype. MSCs are multivalent cells with a superior ability to promote tissue repair by regulating regeneration and inflammation. MSCs have important clinical applications, especially as a more feasible and much safer source for cell-based therapy in regards to stem cell transplant. So these are some of the overarching questions that we try to answer in this study. And is there a specific cell type driving the aging process? And if there are several senescent cell populations that are either specific or universal in aging tissue type, is there a tendency towards a specific tissue type during senescence? And what is the impact of senescence on cells proliferation potential? So here is the experimental design. MSCs were cultured to population doubling. We captured the single cells at 3 different time points representing young, pre-senescent, and senescent MSCs. And there were 3 replicates at each time point. And here is the general steps that we followed to pre-process the data. We had to deal with batch effect issues, which I can explain more about during the Q&A session. Just to give you an idea about the scale of the data, we had around 100,000 cells, but ended up with around 86,000 cells and around 7,000 features after pre-processing. So here is the UMAP of cells. And as you can see, there's no batch effect, and all the replicates are more or less equally distributed throughout the UMAP. We assign cell cycle phase these cells to check whether cell cycle drives any of the clustering, and we see majority of the cells are at G1 cell cycle phase. We ended up with 8 main clusters, and we observed that there are more clusters at the later time points, and that is the senescence state, which indicates that the heterogeneity in gene expression increases during senescence. We then characterize clusters using known cluster specific markers. And just briefly to give you an idea about these clusters, so cluster 6, 2, and parts of cluster 3, they are proliferative healthy MSCs, but there are different subtypes because the markers associated with each cluster was different. And then cluster 5 are cells that are undergoing metabolic stress, and these are mainly T1 cells, and these are the ones that after going through proliferation, cells want to get to a stable phenotypic state. They try to differentiate into different cell types, but because there is no stimulus in the media, they would go under metabolic stress. That would follow by the pre-senescent state, and then from there we will have 3 distinct senescent phenotype, which are oncogenic-related senescent, SAS-related senescent. SAS stands for senescent-associated secretory phenotype, and these are the chemicals these are the molecules that are secreted by senescent cells. And then the last cluster is senescent scapies, which are the most interesting clusters in this study because these cells show some features of senescent cells. At the same time, they do express some proliferative genes as well. So we think they are really, really interesting. Next, we perform an unsupervised trajectory inference analysis to check the similarity of transcriptional profile in these cells. It was interesting to see that the clusters were ordered exactly according to the timeline, which clearly captured the transition process of MSCs from young to an old state. Next, we use SEN package to identify the differentiation potency of cells at different stages of senescence, which essentially scores cells with a Shannon entropy index based on the connectivity of genes in the protein-protein interaction network, which I'm not showing the result here. But what we were mainly interested in was to understand the changes in gene regulatory network at different stages of the senescence process. So we extracted the connectivity matrix and extracted the gene-gene interaction network from our data set, and then computed the Spearman correlation between gene pairs at each time point. One of the main challenges in single-cell RNA-seq data is sparsity, means there are a lot of zeros. And because of this issue, it makes it quite challenging to construct any type of correlation. There's a lot of literature out there proposing different strategies, but I haven't really found any good method that was useful, at least for this data set. So please let me know if you have any recommendations. We then use MCODE to identify network cliques, which are essentially fully connected subnetworks. And you can see that the connections are time point specific. The GRN is less likely regulated at later time points. And this brings me to the next topic that I'd like to discuss, and that is transcriptional noise. So transcriptional noise is the main driver of diversity of cell behaviors in differentiation and disease, reflecting the underlying mechanism of transcriptional regulation. In the context of aging, it is shown that tight control in transcription program is partially lost during aging, resulting into increase in transcriptional noise and heterogeneity in gene expression in a single cell type. Now, the question is how to capture this transcriptional noise given the sparsity and complexity of single-cell RNA-seq data. So to capture the subtle differences in gene expression that are more complex than just mean shift in expression, we used SC-shape to look at the changes in gene expression distribution. SC-shape is a newly developed bioconductor package developed by one of the superstar PhD students in our group. I won't be going through the detail of this method because Melindri will be talking about this package in detail in the following talk. But briefly, through this package, we were able to identify genes that changed their distribution at different time points. This method has different applications, but I'm using it in the context of transcriptional noise. The genes that change in their distribution at different time points were mainly associated with non-coding RNA targets and telomere shortening, which are both known regulators of aging. And these genes were not picked up through the traditional differential expression analysis. So in summary, cell cycle phase is not associated with any specific senescent phenotype, and there are at least 3 distinct senescent states: SAS-related, oncogene-related, and senescent scapies as MSCs age. Heterogeneity in gene expression increases during replicative senescence. There is no preference to a specific cell type as MSC senes. GRN is less tightly regulated at the senescent state, and we were able to systemically identify transcriptional noise through differential distribution testing. So next, what we want to do is to experimentally validate some of the key markers for each of these senescent states, and we'd like to explore different approaches for constructing correlation network in single-cell RNA-seq data, and also explore some cell-to-cell communication between different clusters. With that, I would like to thank members of my group at AIBN, especially Malindri, who developed the SC-shape method, and our collaborator at AIBN, and also IMB sequencing facility at UQ, who did the sequencing. And with that, I'm happy to take questions.
All right. Thank you, Atefeh. We're going to save the questions to the end. So as you guys come up with questions, we're going to ask them all at the end here. Next up, we have Adam Chen from the University of Sydney, and he'll be talking about the TREEKOR package, which is an automated framework for better understanding hierarchical relationships in cytometry data. Take it away, Adam.
Hey, thanks for the introduction. I'm just going to share my screen. Hope it comes up. Great. Hopefully you can see my screen now. So yeah, thanks for the opportunity to present today. I am kind of super glad that the presentation is at this time, because right now I'm talking from Australia, which is now 8 a.m. So it's a bit of a struggle. It's a bit of a struggle to stay up at like 4 a.m., 5 a.m., but here we are now. So today I'm going through TREEKOR, which is a framework that we propose, which enables the extraction of important biological associations in high-dimensional cytometry data through incorporating cell type hierarchies, often overlooked by recent unsupervised clustering workflows. And this is done by quantifying proportions of cell types relative to a parent and relative to the total number of cells. We highlight how these percent parent features can be used to find signal in a case study where percent total had no association. And we also show that percent parent and percent total cell type proportions are important in measuring in both significance testing and multivariate classification. So just before I begin, I'd just like to introduce the group I'm part of. And this is the Sydney Precision Bioinformatics Research Alliance, and we share an interest in developing statistical and computational methodologies. So if you'd like to check out more of our bioconductor packages in analyzing single-cell data, feel free to check out those links there. And I'm currently a PhD student at the University of Sydney, so my advisors, Alice and Jean, are pictured here. Okay, so just to introduce the data that we're working with, so cytometry data, I'm sure a lot of you know about it, but basically cytometry data is a measurement of the properties of cells. And so for example, a sample can be split up into single cells and then marked with markers. And these markers can be fluorescence markers in flow cytometry or heavy metal isotopes in mass cytometry to be ordered to characterize those cells. And to give an idea of the dimensions of the data, with flow mass cytometry, we can have up to millions of cells with up to 50 markers. and scRNA sequencing, where we can characterize thousands of cells with thousands of markers. And obviously, this poses a big challenge with gleaning information from this data. Now, with the influx of high-dimensional data from the single-cell apparatuses, one of the ways for understanding the high-dimensional data is by deducing cell subsets, and this is traditionally done by manual gating or by unsupervised clustering methods, and then measuring the proportions of these cell subsets. Now, in manual gating, this is where an analyst would go through the cells and look at them in 2D scatter plots and then manually annotate the subsets. And this is often a time-consuming process in which, for the many dimensions present in the data, they can only look at 2 at a time. However, with this method, they're able to incorporate the hierarchical structure of the data present by naturally being able to measure proportions of cell types relative to total amount of cells and also relative to apparent population. With unsupervised clustering, methods such as FlowSum or SPADE or SE3 have been used more recently in workflows analyzing high-dimensional cytometry data, and these have been efficient in deducing cell types, but these workflows have naturally overlooked the hierarchical structure of the data, not being able to measure proportions relative to apparent. Now, we present TREEKOR here, and TREEKOR aims to incorporate the advantages of both methods by offering to cluster the cells and then deduce a hierarchy of cell types, and this enables the measurement of cells relative to a parent and also cells relative to all cells in the sample. And this is something that analysts have done as well with either manual gating or attempted to do this using unsupervised clustering methods as well. But TREEKOR makes it easy to extract and quantify these cell type proportions. With the package as well, TREEKOR also acknowledges several hierarchical clustering techniques by offering to use different methods such as average linkage, hierarchical clustering, or HOPAC. And as you can see, this tree here has more than 2 nodes per split, and that's something offered by the HOPAC algorithm. TREEKOR also provides interactive visualization to help view the significance of the cell type proportions with a patient clinical endpoint as well. So now we're just going to go through a couple of case studies to show how TREEKOR has been useful for us. So in a data set where we're trying to uncover differences between latent CMV infection patients, we applied TREEKOR by clustering the data into 100 clusters. And then for each of the cell clusters, we performed a t-test with the patient endpoint for the percent total and plotted the test statistic on the x-axis and using the cell type percent parent and plot the significance on the y-axis. And this is a good way to compare the significance from both views of the cell type proportions. And we highlight here a couple of cell types that we identified. So the CD4 plus T effector memories and CD8 plus CD127 negative T effector memories. And I just want to draw your attention to the top row up here. So if we were to look at these cell types in the data and just measure the proportion of these cell types as a percent total, we wouldn't be able to find an association with latent CMV infection. But that when we look at the percent parent of these cell types, we do begin to see an association with CMV. And this is just to demonstrate that it's important to look at both these aspects of cell type proportions. So further, we benchmarked several data sets as well. So we looked at 12 data sets varying from flow cytometry data sets and CyTOF data sets and a single cell RNA sequencing data set. And here we just plot QQ plots of the statistical significance of each of the cell subsets constructed. And we color them by the type of hierarchy we use. And we basically plot the negative log p-value using the percent parent from using a t-test, and the negative log p-value of the percent total on the x-axis from the respective t-test. And you can see here the dashed line is the identity line. So if we have points above the line in the top right, it means that there was high significance found from the percent parent proportions, and below high significance from the percent total proportions. And from this benchmark, we could see differences in the significance. And highlighted here, the percent parent provided comparatively greater significance. And this basically just highlights that this is an important factor to look at when analyzing cell type proportions in cytometry data. And to further this, we also look at how these work as features in multivariation, as multivariate classification is a problem that's looked at in the analysis of cytometry data. And basically here, we plot the average balance accuracy rates from a 5-fold cross-validation with 20 repeats using the percent total, percent parent derived from hierarchical clustering, and the percent parent derived from a HOPAC tree as features separately. Here we can see differences in the balance accuracies, and so there are some cases which the balance accuracy improves with using percent parent, and some other scenarios where the balance accuracy is better using the percent totals. And here again, different signals are highlighted in the data set by measuring these different cell type proportions and highlighting the importance of being able to measure both. So as a summary, we outline TREEKOR, which acknowledges the hierarchical structure in the high-dimensional cytometry data and provides an easy way to extract both percent total and percent parent cell type proportions. And from the benchmarking and case study, we demonstrate the importance of measuring both. And TREEKOR can be found on Bioconductor and GitHub as well, which we are actively making changes. Our manuscript can be found here as well. And I'd just like to thank the group I'm part of, and particularly my supervisors, Alice and Jean, and collaborators for helping us with biological interpretation. Thanks again for having and listening.
That's great. Thank you, Adam. Appreciate you sharing. So next we're going to have Melindri Demaratni, who's a PhD student from the University of Queensland. And she'll be discussing the SC Shapes package that's currently in development, and it's a statistical framework for identifying differential distributions in scRNA-seq data sets. So take it away, Melindri. Thank you.
Melindri, a PhD candidate at the University of Queensland, Australia. Today, I will be presenting SC Shapes, an R package for identifying distributions in single-cell RNA sequencing data. By profiling the gene expression of individual cells, single-cell RNA sequencing can capture cell-to-cell variation and help us understand how the transcriptome of single cells regulate phenotypic function. However, this also means that single-cell RNA sequencing data are with a high dimensionality, which may involve thousands of genes and many cells. A feature of single-cell RNA sequencing data is increased sparsity compared to bulk gene expression data, where an abundance of cells may have unobserved expression levels due to either biological or technical sources of cell-to-cell variability. As a result, it may be an oversimplification to assume that the expression of profiles of genes in the transcriptome may be adequately explained by a single parametric distribution. A limitation of this assumption may be that some genes are overlooked or modeled suboptimally, as we fail to first evaluate the prevalence of different distributions in single-cell RNA sequencing data. As single-cell data can be driven by outliers, overdispersion, and dropouts, this leads to increased heterogeneity, resulting in multiple expression modes within a given gene. However, a major limitation of existing single-cell RNA sequencing methods is that cell type markers or master regulators tend to be identified by analysis that are driven by changes in average expression rather than focusing on changes in variability. So this limits the ability to discover new relationships or gene expression shape patterns. We address this issue by proposing a gene-specific framework for identifying differential distributions. In our framework, the input is the matrix of UMI counts, and we filter out genes not expressed in at least 10% of the cells. Then model-based normalization is done for normalization of cell-level covariates, such as batch effects and sequencing depth differences. We then model the UMI counts using the four distributions: Poisson, negative binomial, zero-inflated Poisson, and zero-inflated negative binomial. So all these four distributions are special cases of the zero-inflated negative binomial distribution. Hence, a Kolmogorov-Smirnov test is first conducted to identify genes belonging to the family of zero-inflated negative binomial distributions. Then the UMI counts are modeled using the four distributions with log-link function. Model selection is done by calculating the Bayesian information criterion, where the model with the least BIC value is selected as the best model. Further model adequacy tests are done by calculating the likelihood ratio test statistic, and we also test for the presence of zero inflation for the zero inflated distributed genes. Based on the results of the model adequacy test, each gene is then classified as following one of the four distributions. To test the performance of this model, we conducted a simulation and also applied this framework to real single-cell RNA sequencing data. We conducted a simulation study to assess the ability of our framework to classify gene expression distribution. Using model classification and parameters estimated from the 10x PBMC data, we simulated Poisson, negative binomial, zero-inflated Poisson, and zero-inflated negative binomial distributed genes for a range of sample sizes. The graph shows the percentages of correctly classified genes. So we can see that our framework can accurately identify the correct model distribution for each feature with an accuracy of 85% or above for all four distributions across the range of sample sizes tested. For framework development and testing, we use single-cell RNA sequencing gene expression measurements collected from mice on two tissues, adipose and muscle. The primary dataset used describes one of the most common biological designs used in single-cell RNA sequencing experiments, where single cells are profiled from three different treatment conditions: young, old, and another group treated with metformin. However, perturbations to single cells can take on other forms, and therefore we selected multiple other experimental designs to demonstrate how our framework can also identify useful biological results with different scales of complexity present in the data. By applying SC shapes to single-cell RNA sequencing data from multiple experiments, we were able to reveal new insights that would have been otherwise missed by standard single-cell tools. When applied our framework to single-cell RNA sequencing measurements on the metformin dataset, we see that while majority of the genes follow a negative binomial distribution, at least 50% of the genes in muscle tissue do not follow a negative binomial distribution. Next, we wanted to see whether accounting for known biological knowledge, such as the cell types present, as well as other covariates like the mouse ID, would result in a change in the classification of genes into one of the four distributions. We found out that after accounting for known biological variation, majority of the genes followed a Poisson distribution with a handful of genes following zero-inflated distributions. So this provides evidence that in fact, not all the genes follow a negative binomial distribution, as well as not all the genes in the transcriptome follow a single distribution. We also compared our framework against some of the widely used tools for identifying differentially expressed genes, namely EJA and SCDD. And we found out that among the uniquely identified differentially distributed genes by our framework were the transcription factors Forkhead BOXO3, which is often referred to as the longevity gene. And we also see interferon regulatory factor 3, which is a novel inhibitor of cellular senescence and inducer of cell growth inhibition, to be detected by our method. So one of the main advantages of our framework is the ability to detect zero-inflated genes. And zero-inflated genes are important as potential underlying biological mechanisms for zero inflation might be transcriptional bursting or stages in the cell cycle or even activation status of cells. However, more research has to be done to confirm these hypotheses. As the next step, we are currently exploring the possibility of extending the differential distribution framework to other types of single-cell omics data, for example, single-cell ataxic. We're also looking at the possibility of incorporating other distributions that may be useful for modeling heterogeneity, for example, like mixture model or even Dirichlet processors. And by extending our framework to include other types of single-cell omics data, we hope to test whether particular classes of genes have specific distributions. In summary, here we present novel statistical framework for quantifying gene expression heterogeneity by modeling gene expression levels under the differential distribution pipeline. So compared to existing tools, SC-Shapes is advantageous because it can handle excess zeros in single-cell data, adjust for covariates, as well as compare multiple groups of treatment conditions. In conclusion, our case studies highlight that distributions are gene-specific and one distribution does not fit all. We also found that a Poisson error model is actually sufficient to explain the variability in the expression of genes with a housekeeping function. And our case studies also open questions about the plausible biological mechanisms that may give rise to biological phenomena such as transcriptional bursting. So we have implemented our modeling framework as an open source R package, which can be downloaded from our GitHub. And our package is also now available through the development version of Bioconductor. So finally, I would like to thank all my supervisors and everyone.
That's great. Thank you, Melandri. So our last speaker is going to be Shiji Zhang. And he is a postdoctoral fellow at Johns Hopkins Bloomberg School of Public Health, who will be discussing the tricycle package that infers cell cycle position for single-cell RNA-seq datasets.
Hello, everyone. My name is Shiji. I'm a postdoc at Johns Hopkins. I'm happy to share our recent work in universal prediction of cell cycle position using transfer learning. We know that the cell cycle is a fundamental biological process in almost eukaryotes. It's highly conserved and continuous process in nature, and is also tightly regulated, as this regulation is associated with many diseases, such as cancer or Kabuki syndromes. The time to complete a full cycle varies greatly from one cell type to another, even in the same organisms. The emergence of the single-cell technology makes it possible to analyze the transcriptional dynamics of a cell cycle at a large scale and fine resolution. However, there is no universally applicable approach to infer the continuous position in cell cycle with high resolution from single-cell RNA-seq data. That motivates us to develop this new tool. Our method actually comes from an observation that the first two principal components of the cell cycle genes always forms a circle on the dataset of which the mean variation is pure cell cycle. This circular behavior has also been observed in the t-SNE or the UMAP embeddings too, and it was reported by a lot of other papers. The question comes down, why do you see this circle? So there is a relationship between the principal component analysis and the Fourier analysis. We know that for a circular matrix, it can always be diagonalized with the discrete Fourier transformation. To explain it in a very simple way, we simulate a bunch of genes with periodic functions, and here we show you the expression of two genes. If we don't know the time point, we can just pretend that we don't know. We permit a time point. For both of those two data sets, if we run the PCA, we will get exact PCA. And we can use this angle formed by the top two PCs to successfully recover the original time point. This was kind of ensured by some important characteristics of the cell cycle genes. First of all, the G2M genes always has highest expression, and the G1S genes has a low expression. And this corresponds to search of the direction of the PC1. Another thing is that all the genes, all the cell cycle genes over cell cycle only have one peak. And the third one is at least 3 different peak locations I found during the cell cycle. So we first just do the PCA on some of the data sets, like the two of them shown here. They are 2 independent data sets. And if we run the PCA on the cell cycle gene, we can see that the weights of those genes of PC1, they are highly consistent. And for both of them, we observe this circular relationship. If we use this angle to measure the cell cycle position, we see we see that the expression dynamics over this inferred the PCA theta here, they are quite consistent between those two data sets, as well as the dynamics of the total UMIs. And more importantly, the differences between the peak and the bottom here corresponds to 1 at log 2 scales. And this corresponds to the fact that the relative amount of DNA per cell kind of is doubled for the cells in the G2 and M phase compared to the cells in the G1 phase. However, this simple approach won't work in a lot of other data sets. Sometimes it just fails. For example, we use another data set to show that. So for this UMAP, on this UMAP, we can see that the mean variation of this data set is the cell type differentiation. The most multipotent cells are kind of here, the ductal cells and NGN3 low EP cells. If we just do the PCA on the cell cycle genes, we see that the PC1 still reflects the differentiation rather than the cell cycle. This is caused by the fact that most of the genes participate in multiple biological processes, and we don't get an ellipsoid when there are other source of variations other than cell cycle. Another interesting fact is that if we subset the original data set to one of the cell types, such as the ductal cells, and run the PCA on the cell cycle genes, we can still recover this ellipsoid circle here. But this solution won't be applicable to a lot of data sets we have, because we don't even have a good cell type label. To circumvent this problem, we resort to the transport learning idea. It was first proposed by Penn, and it was validated by the Stan O'Brien, and it has been demonstrated to work for the single cell transcriptomic data. And that comes down to our tricycle method. We learn a reference on the data set of which the mean variation is cell cycle progression. We use the mouse neural sphere data set here, and we project the cell cycle embeddings into the new data. So in this figure, we show you that the projection works quite well on those 4 data, especially if we focus on the second column, the mouse pancreas dataset. Previously, the PCA fails on this dataset. the projection works perfectly on this dataset. We further validate the tricycle inference on the gold standard dataset, including 2 Fuji and 2 FACS. I show you one of the Fuji dataset here. So the first panel here is the Fuji measurement of 2 proteins. And the second one is show you the good alignments between the Fuji cytotime and our cell cycle positions with a circular correlation to 0.7. And more importantly, the accuracy of the cell cycle compares favorably to the golden standard experimental assays, which generally require specialized measurements in specifically constructed in vitro systems, as shown in this as proved by these R-square values here. We further applied the tricycle on the big single-cell RNA-seq atlas data, which has more than 4 million cells. It is being shown that the tricycle has great generalizability, especially this dataset is a shallow sequencing dataset. The median of the total UMI numbers is only about 500 and is quite scalable on those 5 million cells. We successfully identified the most proliferative tissues and cell types in these cells in this single-cell RNA-seq atlas data. We further assess the stability of the cell cycle position by considering using the downsampling and randomly removing genes from the reference data. For both of them, we have shown that the tricycle is quite robust across different scenarios. In summary, we have used the tricycle on 26 datasets, and it has been shown quite generalizable across species, including mouse and human, and across different experimental platforms, which is quite amazing. And it is also generalizable across tissues and cell types. It has been shown that it outperforms other similar cell cycle inference methods in terms of the accuracy and speed, and is quite scalable. On the dataset of 50,000 cells, it only takes about 1.48 seconds to run. And the most important thing is it is comparable to golden standard experimental assays, including the Fuji and FACS. But I also want to mention that since the motivation of the tricycle is to model the dynamics over cell cycle, we do not recommend using tricycle on the non-cycling dataset. It is now the method is described in our preprint, and the function is available in the bioconductor package tricycle. And I would like to thank all the people who contributed to this project, as well as our funding source, the CZI Seed Networks. And of course, thank you so much for your attention.
All right, great, thank you. So now we're going to open it up to some questions. We have a question here from Kelly. Let me put that on the stage here. Do you think tricycle could be used in a similar manner to infer circadian rhythm cycles? Shijie, go ahead.
I'm not so sure about that because I think we have to check the behaviors of the gene expressions. There are multiple characteristics to ensure that the projection works for the cell cycle genes. So yeah, that's my answer. So I'm not sure.
Okay, and then we have another question here from Christoph. Could you comment on what a non-cycling data set is?
Yeah, so if you have a data set with all the terminally differentiated cells, so mostly, I mean, the majority of the cells are non-cycling. That's what I call the non-cycling data set.
All right, thank you. I have a question for Atefeh and Melindri. So Melindri, in your talk, you had an example of MSCs. I saw that in the bottom right corner of one of your slides. Was that one of Atefeh's data sets? And if so, like how did you, Atefeh, end up using SC shapes to help you identify your results? Like, what was the before and after, I guess?
Yes, that was Artie's data set. Artie, would you like to?
Yeah, so what I was really interested in is because, as Melindri explained, some of these differences that we wanted to check in terms of transitioning of cells from young MSCs to age or senescent MSCs, we really couldn't capture them through the traditional differential expression analysis. And then because of the features of single-cell RNA-seq data, obviously from the pre-processing and normalization, you assume that the gene expression distribution is similar for all the genes. So we really wanted to test and try and check whether that holds true for the data set that I was using. And we realized that obviously I showed a table at the end of the talk. We had different categories of genes that were changing the distribution at different time points of this process. So that was really interesting for us. And at the same time, because the models that Melindri actually put in the framework, they were also able to capture the zero inflated type of the distributions. And as I said, because of the characteristic of single-cell RNA-seq data and because it's sparse in so many zeros, it was really important for us to be able to account for that variability as well. So as Melindri explained, so, I mean, there are talks in the context of transcriptional bursting, as obviously transcription is a stochastic process. It goes up and down. So we were really interested to check whether we are able to capture that behavior in the MSC data set as well.
Interesting. We have another question here from Matthew. Melindri, are there genes that don't appear to follow any of the distributions that you've considered?
Yes, that's correct. There are genes that don't follow any of the 4 distributions. So here the assumption we made is that most of the heterogeneity can be explained using the 4 distributions. So that's why we first do a Kolmogorov-Smirnov test, because since they all belong to the same family of zero inflated negative binomial distributions, and so we only subset those genes. So we didn't actually want to include any other distributions because one thing is, if you introduce continuous distributions, so then we are kind of changing the like the structure of the data, going from discrete to continuous. And also the more distributions you have, the harder it will be to biologically interpret what these distributions actually mean.
Interesting, thank you. Adam, I have a question for you. On your TREEKOR dataset, you were talking about parent percent. Is that a parent percent as in from your hierarchical clustering, or is that an actual real parent gene of like parent sibling genomic data?
Yeah. Oh, good question. That's from the hierarchical clustering that's done. Although it is inspired by a manual gating tree, and so if you did have a hierarchy of cell types, for example, yeah, you can have CD4 T cells, and then naturally you'd have a child proportion like a T-conv cell. Then you can put in the tree defined by that, and so the percent parent is just measured according to the cell tree. And for the case where you don't have that tree, it will just get the tree from the hierarchical clustering. Yeah.
Okay, cool. Thank you. Well, if there's no more questions, then we are at the end of time. We're a minute over, but yeah, very interesting. Thank you, everyone. Really appreciate your talk.